Okay, so let's start. So, good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome, and thank you for joining us, the 32nd seminar and Valencia Day. I am so glad to meet many SynBYSS people Sunday and Monday in person during the SEED conference. Of course, actually the best synthetic biology conference, I would say, in Washington, D.C. And today's speaker actually coming from that conference after the presentation. And also I am right now in New Orleans because I'm also attending another, because I organized 2 conferences this week, crazily. And that's the SIM conference in New Orleans, very interesting city. And actually this is my 7th in-person seminar or conference this year. And this has been so great, but I want actually more. And actually I escaped from the ONR grant meeting, and actually I cannot to go back. Otherwise my program manager will not be happy. But personally, I'm on vacation to take 70 days of trip, mostly in Europe, starting mid-May, to give many seminar and invited talk. Still, our SynBYSS virtual seminar must go on. So I'm connecting and chairing the seminar from my hotel room, New Orleans, after listening to jazz until midnight yesterday. That is crazy night yesterday. So, okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Alfonso Jaramillo. And he's, of course, a professor at University of Valencia in Spain. He doesn't need any introduction because we all know him. He's, of course, an editor-in-chief for BioDesign Research Journal, a AAAS partner journal. And that's a very exciting journal. So if you have the paper, to submit to that journal. He was my first collaborator in 2006 when he was a professor in Paris at Polytechnic, where I visited long time ago. And of course, I'm glad to see him again in this format. And it's very good to see again, virtually, unfortunately, but probably in near future in person. So Alfonso, the virtual podium is all yours. And thank you so much for your time, even though your time right now is not that convenient because that's a late afternoon. Thank you.
Thank you very much. And thank you for this honor to be here talking to you. After all these other pioneers, I hope that I can be also useful for the young people. So let me just explain a little bit my strange career path. That I did my PhD in particle physics, in theoretical particle physics, so like Sheldon in this Big Bang theory. Okay, so exactly same stuff. So, but then I changed to in biology. Even without having any knowledge of what is the difference between DNA and protein. I thought the protein was in the meat, but not in the salad, something like that. So that was my knowledge of biology when I moved to biology. So, but still, despite of these holes, I got postdoctoral offers, and then, in fact, I got all the offers I applied. And then I was lucky that I went to work with a professor that she's Shoshana Bodak. Did you know her? Because and then when I went to her, I could then find a path in the future of that is a long-standing path that is to find general principles for the design of in biology. So at that time I was working on protein design, but not a particular protein design of just a particular protein. No, in general, general methodologies to enable the design, a general purpose design of proteins. So then it was more structural based. So this means that we use the atomic coordinates of the proteins, and then I did a software to simulate the dynamics and then all the evolution in particular, all the mutations. And so then that theme has been, I followed that throughout my career. So during my postdoc years, and I did 4 years in a postdoc in total, and I was doing that. So of course, I changed from physics, so I had to just focus in one single thing. But then after that, when I had my first junior professor position at Polytechnic, and then I could start changing a little bit the subject, and then instead of doing the design of proteins, I thought that it would be good to do the design of networks of proteins, okay, and then gene circuits. And I also got interested in the design of RNA. Can we apply the same concepts that are physics-based? Because of course you are biased, because I was a physicist, so then I thought where I could contribute with my knowledge the best, given that most people have many more years in advantage than me. So I thought that this is interesting. I mean, to tell the people this story, a little bit of how I've been progressing on the concepts, because I mean, sometimes you have to reinvent yourself, and how I reinvented myself a few times, and then, but always the same subject of doing and following always one single goal that is a general principle for engineering and de novo in biological matter, let's say. Okay, so that's either de novo design of RNA, de novo design of circuits. So that's probably the most used word in my papers, this de novo thing. And so, and then and for young people, I will advise that and to follow always in this very focused principle, but that you could implement it in many ways. I mean, I could implement it with proteins, I could implement it with RNA. Now I'm implementing and doing, and you can also some directed evolution with phages, and then and also some this engineering an artificial and a neural network with bacteria, and doing deep learning with bacteria. So instead of the normal deep learning people do, I mean, with GPUs, you do it with E. coli. Okay. In fact, I'm looking for people for that. So just I have a postdoc position for that, and then I also have a postdoc position for evolution with phages, just in case. And so in beautiful Valencia, Spain, which is a nice place. I'm biased, but okay. So. So then, having said that, I think that for always I like to provide a lot of advice, because to the people, to young people, and that it's always, you never know what's going in the future, but it's important to keep your focus on some principles, not to sell your soul to their funders or things like that. Sometimes the worst thing can happen is when you apply for a grant is that you get the grant. So you have to be careful and that apply to things that are within your values, scientific values, I mean, and then be consistent with that. Otherwise, you may get trapped to do things that really don't excite you very much. So it's very important to do things that really passion you. And okay, so so then that's, I've been then having many positions. I just started in Valencia, so that but I have to say that the most important thing is the collaborators to convey your passion to the younger people, and then because these are the real people who are doing the everyday experiments. So then, and these are the legacy you have when you just pass all your motivation and your excitement and some knowledge maybe to the new generations. Okay, so I'll be very excited to listen to Eric.
Also, fantastic, Alfonso, a great talk with the wonderful advice for young people. Thanks. I mean, I am amazed by your talk. So of course, I mean, I was excited because of you, not because of Paris. The 20 years ago, whatever. Also, your unique path shows you are a true pioneer, and I also really want to identify the governing principle or law of biology, like Newton's law in physics or energy conservation law in chemistry or thermodynamics. But that might be holy grail, I mean, to find governing principle of biology. But I believe Eric or young people will find those kind of principles in near future. So I'm very optimistic. And thank you so much for your visionary advice and on the talk. And this is truly amazing. Thank you.
Thank you to you.
Yeah, thank you. Okay, so now the main speaker of today with a longer introduction. Eric Young is assistant professor of chemical engineering at Worcester Polytechnic Institute. His research objective is to understand and engineer novel microbial systems that improve the human condition, focusing primarily on the unique phenotypes and metabolism of fungi, the most challenging, microbe. I never touched this one because it's so damn difficult. But he's brave enough to take on this project. And because synthetic biology is so underdeveloped in this organism course, his team and others are addressing multiple areas of need, including genetic design aid, method development, genomics data acquisition and workflows, modular part collection, pathway engineering. This research program informs his educational goal to train the current and future workforce for an economy shaped by engineering biology. Even if he's so young, he was so very passionate about the education of younger people. He received numerous awards, including NSF Career Award and also Massachusetts Life Science Center Building Breakthrough Award, to name a few. Eric, received undergraduate degree in chemical engineering and biological engineering from the University of Maine. He received his PhD in chemical engineering from the University of Texas at Austin, supervised by Dr. Hal Alper. He's another, big guy in this field, as an NSF graduate research fellow. That's one of the prestigious, award for the graduate student. He completed postdoctoral research at MIT, contributing to the development of the MIT Broad Foundry, supervised by Chris Boyd, another big guy, in synthetic biology. He's a true rising star, already demonstrating his excellence and giving a lot of talks, including a talk in Portugal, where I was also invited. So as you can imagine, he is truly a rising star. I believe he should be right now associate professor with tenure or even full professor. And so if tenure letter writers watch this video, please consider writing one of the strongest letters for him, because he deserves that strong letters. So Eric, please take away, and thank you so much for your time in this kind of crazy week with a lot of conference happening in multiple different places. Thank you.
Thank you for the wonderful introduction, Taesuk, and thank you for the advice, Alfonso. Such a great opportunity to speak to you today. Let me get my slides up. Let's go to here, and then do this first. Get to see a picture of my desktop, and then you should be able to see the slides.
I see your note slide as well. You may want to display setting and then kind of flip.
Yeah. New share. Screen 2. Yeah, it was on screen 1, not screen 2. That should be it.
Right, perfect.
Perfect. Okay. Always, you know, still we're almost 3 years into this, and we're still a little bit awkward getting it up on virtual presentations, but that's okay. As Tae-Suk mentioned, I'm assistant professor in chemical engineering at WPI. And today I'm going to talk to you about our work in fungal synthetic biology. And we're going to touch on a lot of the broad subjects that Tae-Suk so clearly introduced what we're interested in. So I'm also from a fairly beautiful part of the world. I'm from the state of Maine. That's where I got my undergraduate degree. And I've always been interested in fungi because they are essential in nature and in culture. One of the things that they do is a very common thing in Maine, which is wood. They break down biomass. It's kind of how I got started in metabolic engineering and synthetic biology, is dealing with the breakdown products of lignocellulosic biomass and trying to convert those into products. But as I've spent a lot of time, you know, reading and learning about fungi, you really realize how much more essential they are besides just as saprophytes for recycling biomass. They connect things in the soil microbiome. I'll talk a little bit about that today. They make natural products and fuels, which are really important for a lot of the build with biology efforts we have going on in the world today. And they make nutrients and foods. One of the things driving not only foods we've had for thousands of years, but also foods we are now making as meat replacements and so forth, use products of fungi. And just as a note, because I think it still needs to be said, very basically, you know, fungi are eukaryotes. And when we talk about yeasts, yeasts are fungi. They just have a unicellular lifestyle. And this is just showing the general fungal tree divided up into sort of 3 groups, which are basal fungi, ascomycetes, and basidiomycetes. I'll be talking about mostly the ascomycetes and basidiomycetes today. But what this shows is that yeasts are a lifestyle that different organisms across the fungal tree tend to choose, not a family. And this is in you know, taxonomic terms called not monophyletic. And so we're interested in yeasts and fungi in my group, and the organisms that associate with them. And so what we are basically trying to do, kind of what Alfonso mentioned, what our overarching goal is to enable fungal research at the intersection of synthetic biology, systems biology, and metabolic engineering. We really need these diverse tools and other multidisciplinary tools to bring a lot of these organisms we're interested in, perform engineering for an application. And so when we're thinking about the type of engineering we want to do, everything is really enabled by the genome sequence and transcriptomics, if we can do it. And that's really from the discipline of systems biology and bioinformatics, and also parts for programming these organisms that many people work with. And so we do a lot of part development in my group. And the applications we're kind of looking for are to make cell factories and also to program microbiomes. So 2 different applications that you might use engineered cells for. These are the ones that we are excited about. So obviously this is a lot of, like, we contribute in different areas of need, as Tae-Suk said in my bio, for addressing how to get from a yeast that not many people have worked with to something that everybody could work with, and to try to make people less scared about moving into this wonderful you know, branch of the tree of life. And I'm going to emphasize 3 areas that we really have contributed to early on in my lab's work. One of those is sequences and sharing them with others. The other one is developing what we like to call platform hosts for cell factories. So we like to choose organisms that already have a natural metabolic network set up for us to produce molecules of industrial interest. And one thing that we've been working on lately is harnessing the emergent properties of microbiomes, which is a little bit different, but it does still incorporate a lot of what we have been doing in fungi and the organisms that associate with them. So we're going to step back a bit and talk about this first section about what I mean by sequences and sharing them. This is a big part of the field of synthetic biology. We like to compose everything of units of DNA. And we also like to use next generation sequencing from the biologists to look out in nature, collect a great deal of sequence information that dictates biological function, and use that to program biology. So next generation sequencing, as many people point out in many talks, has really been driving synthetic biology, because the more data we have about DNA sequence, the more ability we have to know what works and to source it to program biology. A division of, or like a subsection of next generation sequencing is whole genome sequencing. And this is really useful for a lot of the organisms we work in, because those whole genomes are, maybe not been performed before or are low quality. And I want to spend some time in talking about how this works, because it kind of informs the first bit of work we were able to do in this area. So the first challenge is actually isolating DNA from the organism you would want to sequence. For some organisms, it's very easy. For others, it's difficult. Then the second step has to do with library preparation, where the genomic DNA is treated enzymatically or mechanically in order to be prepared to go onto a sequencer. So it has the chemical decorations necessary for the different sequencing technologies to work. And that creates computationally now some data, either in long reads of DNA over kilobases, or short reads, which are only a few hundred base pairs, that need to be stitched together by software into what we call contigs in the assembly process. And those things are then annotated to give us a whole genome from which we can extract information or use for different purposes. And we really got started on this project because we really wanted to get accurate whole genomes for a lot of the fungi that we work in. And we really wanted to get an idea of whether we were performing the engineering we intended. And so we really want to be able to, in an organism that we've engineered, and maybe even heavily engineered, find all of the plasmids, genome integrations, deletions, and mutations we may have done to that yeast or fungi, and get an accurate picture of whether we did what we said we were going to do. And this is kind of informed by a body of literature as well that was showing that many unintentional edits occur. Many of you may be familiar with unintentional edits that occur by CRISPR editing, but a lot of other unintentional edits can happen. Multiple insertion events can happen when you're integrating by a homologous or combination or non-homologous end joining. A lot of yeasts are very flexible in their chromosomes, particularly Saccharomyces cerevisiae, which is the most common yeast that many people are familiar with, can rearrange its chromosomes quite easily. And there's actually a whole field of research led by Jeff Boeke that shows how those chromosomes can be rearranged synthetically, which is quite interesting. And as well, you can actually have unintended insertions of plasmid or cloning host DNA. And so, these also kind of mark our interest. Not only do we want accurate whole genomes, we want to accurately resolve engineering. And it turns out when we first started doing whole genome sequencing on some of our engineered Saccharomyces cerevisiae strains, so very basic yeast, we could not get back what we had put in the sequencing data. So we would expect from this yeast that has a carotenoid pathway in it and a couple of plasmids, we would expect to get back in our whole genome sequencing assembly this full pathway and each plasmid. But in fact, we get the sequence back that's fragmented across multiple contigs, and we even get missing parts that aren't there in the sequencing assembly. So we engaged in a large effort. My first graduate student engaged in a large effort as he was building out our tools for working with a lot of yeasts, also working on the computational sequencing side to debug this process and figure out how we can get accurate engineering out of a genome. The first thing that Joe did was to tune the library preparation to increase plasmid representation. So if you look at this display here, you can see that what's really dropping out and what really are struggling are the plasmid sections, which is the right-hand side of this slide. And so we tuned our preparation of the DNA that we put onto the sequencers to represent the plasmids. And we were able to get more plasmid reads. And that increased our ability to assemble plasmids. But really what we started to see was that we had enough data. The assembly software wasn't doing what it should be doing. So we tried many of the assemblers that were out there. And while these are great at getting whole genomes by the metrics they were measured against, they really didn't perform that well when we were looking at our engineered pathways and plasmids in our yeast. And we were kind of, disappointed in this, but it's kind of an example of the tool does what it's designed to do. Luckily, one of the newer tools that really used the nanopore long reads, Fly, did a very good job. And so we were able to use this assembler going forward to get accurate genome sequences. But one of the things that we noticed was that when it started reporting back what the plasmids were, it was actually 2 or more repeats of the plasmid sequence. So it just kept going around and around and around. It wasn't connecting it. And this was an issue because, again, Fly is designed to assemble nanopore reads, and it's actually designed to assemble chromosomes because it's designed to assemble eukaryotic genomes. So it didn't really recognize that it should close the loop on the circular plasmids. And so we had to use a bacterial genome assembler unicycler and send those plasmid sequences to that software so it could assemble an accurate single copy plasmid. So that led us to a hybrid genome assembly pipeline where we take nanopore reads and Illumina reads and feed them both to Fly and unicycler to accurately resolve DNA signatures across chromosomes and plasmids. And this is, as far as we can tell, the only way you can do it right now. Maybe as software assemblers advance, you could do it with a less hybrid approach, but really we're taking advantage of the benefits of long reads and short reads, as well as the ability to assemble chromosomes with Fly and the ability to assemble circular DNA with unicycler. And once we do this, we can look at a variety of engineered strains that we've made, and we can accurately assemble all kinds of different pathways, all kinds of different synthetic biology, yeast plasmids, low copy and high copy. And we can also look at, finding a lot of gene editing elements like Cas9 and so forth in these strains. So our tool can accurately return the engineering that we intend to put into the yeast. We had a couple of edge cases, but these turned out to be actual unintended edits. So we did have a representation of some of those unintended edits I was mentioning earlier. We expected to engineer the non-conventional yeast K-FAFI in chromosome 2, but we actually found the pathway in chromosome 4. And we backed this up with PCR confirmation that this was actually where the software tool said it was. And then we also engineered Uroia lipolytica, and we were missing a big chunk of a plasmid. And we thought, oh no, it's a recurrence of some of the issues we've been seeing earlier where the sequence is dropping out of the plasmid. It turns out when you do a PCR conformation of the plasmid, you do find that it is truncated. So it is in fact missing half of that. So we actually weren't very good at engineering a couple of these yeasts, but that actually turned out to benefit us because it shows that our tool diagnoses these unintended edits that occur. So we've built a tool for what we wanted, what other people haven't been doing before, which is getting engineering out of a whole genome sequence, and particularly out of a whole genome sequence of yeast. And the bigger question is the rest of the genome good quality? So have we done something where we made a tool that works for what we want, but sacrifices whole genome quality? And the answer is no. It does not sacrifice whole genome quantity. So these charts may be a little bit unfamiliar to folks who don't do a lot of whole genome sequencing, but what we have here on the left is a whole genome completeness score for a bunch of different Saccharomyces cerevisiae yeasts and non-conventional yeasts, showing that we can get highly complete all of the essential genes, highly complete genomes. And on the right here in the green heat map, we're showing that these sequences are also contiguous. So we can align everything to all 16 yeast chromosomes, all 6 chromosomes of Uroia lipolytica, and all 4 chromosomes of K. Faffi, and the aligned bases are somewhere in the high 90s. And so the genomes that we get are nearly complete and contiguous, so they accurately reflect like chromosomal resolution and plasmid resolution. And this is really exciting because that means that we can go and look at our yeast genomes and annotate them with telomeres and centromeres, the features of chromosomes, and brightly light up where we're putting our engineering. And this is another picture of where we've inserted a pathway into scaffold 3 here, and you can see the red square is showing where the pathway is. And in the smaller scaffolds, which are the plasmids, we can also show where the engineering is. So not only can this tool return the engineering back, be a quality control from a whole genome level for what we're doing in yeast, it can highlight engineering if we're looking for it. So the final pipeline kind of looks a little bit like what I've shown before, except we've attached, we have a functionality where you can add in engineering sequences or signatures you want to look for and blast those against the final assembly and visualize them in different ways. And we also showed in the paper that came out about this that we can resequence non-conventional yeasts, particularly one of interest to the field is Uroia lipolytica, and we can get a much more contiguous, much more complete genome out of the tool. So I think this is broadly useful. One of the other use cases we showed is that we could pull engineering out of a metagenome. So if you were to sequence, several dozen bacteria and yeast, a community maybe, you could, if you had enough reads, you could pull out the integration in the yeast genome and the plasmid, and that we showed that even up to a 1 to 1,000 dilution of the engineered organism. So this tool, I think, is broadly useful for any type of application where you really want accurate resolution of engineering. It can be used, like we use it for quality control of what we're doing, or it can also be used to analyze whether there's engineering in unknown samples. It's on GitHub, it's available. We work with and collaborate with several groups around the world who are now using this to debug and assemble genomes. And let us know, feel free to use it. Here's the link for it. And if you would have any issues with it, we monitor the issue tracker as well. So please feel free to reach out to us about this. So that kind of concludes my section on why sequencing is so important and why we've been contributing in it, is to get accurate resolution of our engineering and to get accurate whole genome sequences of some of the yeast we might want to work with. And that sort of brings me into my second section of my talk, where we're sort of dealing with developing what we call platform hosts for cell factories. And these are not organisms that a lot of people around the world work with, and I'll explain those a little bit, but I have to have a little bit of physics here. Since Alfonso does come from a physics background, this is a diagram of our solar system, right? And for many centuries, our solar system was the only thing we could study. So we had to infer whatever we concluded about our solar system was perhaps very general. There was a mission a few years ago called the Kepler Space Telescope, which with a variety of methods was able to analyze the distribution of solar systems in a region of our galaxy. And it found that our solar system was in fact the exception and not the rule. There are many other different arrangements of planets and sizes, and our solar system is rather strange. And I really like this analogy because we are used to, in our field, of taking all of the sequence data that we obtain and putting it in a chassis of a model organism like E. coli or Saccharomyces cerevisiae, when in fact E. coli and cerevisiae are sort of the exceptions and not the rule. They are, E. coli is from the intestines, and cerevisiae is a domesticated yeast that's great at making sugar, converting sugar to ethanol. And so, I like to think about this. Why are we putting all of this effort into making yeast do something other than convert sugar to ethanol, or making E. coli do something other than participate in the activities of the human gut microbiome, when there's all kinds of other organisms out there that have different functions that may be a better choice? And we can learn a lot more about biology and find out that sometimes the biology of E. coli and Saccharomyces cerevisiae is an exception to most microbes and not the rule. So this is what really inspires me to move. There's also a move into other interesting organisms. There's also a really strong metabolic engineering argument for why you would want to do this. So my background is in Saccharomyces cerevisiae. That's what I spent my PhD and my postdoc really working on. And in my postdoc, I developed massively parallel iterative combinatorial pathway engineering in yeast, where we take all kinds of different parts, we hook them up to a pathway, we make hundreds of different versions and test the function of all of those different versions and try to learn how to make the pathway better. And this really works. That was really the exciting result of all of this, all of these articles, is that it does in fact work. We were able to produce almost a gram per liter of itaconic acid in Saccharomyces cerevisiae, and that was a few fold more than others had yet achieved. But in the preparing that data for publications, I was reading about the native producer, Aspergillus terreus, and this is in fed batch, so it's kind of gaming the productivity of the strain, but they're able to achieve several orders more magnitude of just the natural producer. So I became very discouraged of why would I spend so much time and effort and money transferring the genetics from Aspergillus terreus to Saccharomyces cerevisiae and cost myself several orders of magnitude. I'm picking on myself here, right? But there are a lot of examples that out there from others that I won't mention, where this kind of function between where you've gotten the genes from and done all of this really great metabolic engineering and synthetic biology and put it into a model organism, and it doesn't function as well as where you got the genes. So this was a very personal result. Again, a great achievement of synthetic biology and showing combinatorial pathway engineering can work. But again, from the standard of what we apply in sort of the metabolic engineering circles of how much are you making, this kind of falls short. So I thought this was a very strong motivation to found my lab again on moving away from comfortable laboratory conditions and comfortable organisms and into other production environments and other organisms better adapted for the intended the purpose we intend. And so one of the stories that we encountered very early in my lab, in sort of looking for different examples of this, was the carotenoid pathway from the red yeast Xanthophyllomyces dendrohus. And what had gone on there was the pathway was taken from this yeast and put into E. coli and Saccharomyces cerevisiae to make astaxanthin. And the story was kind of the same there. The native organism makes much more astaxanthin than the engineered strains of model organisms. So a little bit about this organism. We don't really like to call it Xanthophyllomyces dendrohus all the time. We just call it red yeast. It's a really promising terpenoid cell factory, and this is because the production of astaxanthin is part of the terpenoid pathway, which is an iterative buildup of isoprene units and other units composed of isoprene into progressively larger molecules, which can then be functionalized into a variety of natural products. And we're really excited about this organism. It's genetically tractable, actually. There is some work out there that shows that it can be engineered. This is kind of what it looks like under the microscope, and this is what it looks like in colonies. It's naturally red and orange. And so, you know, we're really excited about, instead of taking the genetics out of this organism, can we develop tools to work in this organism to improve its function of the carotenoid pathway. So just a little bit of basic science. Where is red yeast? So here is our fungal tree again. Here's our three major divisions of fungi. And here's, you know, these species are familiar to many who work in yeast, but these are many Ascomycete yeasts and filamentous fungi that people generally work in. And I've bolded Saccharomyces cerevisiae there in general. There's also a few groups around the country and the world that are moving into Basidiomycetes, particularly Rhodosporidium torulioides, and in the past, this Phanerchaete chrysosporium organism. And then there's also folks like Michelle O'Malley and others in her trainees that are moving into basal fungi and studying the biotechnological relevance of those. And when you kind of look at what's going on in the field and you read about this really interesting red yeast, you find that it actually exists in an understudied position in the fungal tree compared to the other organisms. So we think that we have this unique combination of industrial relevance and you know, scientific question marks, that this is worth further investigation. One of the reasons why it isn't more widely used to produce a lot of terpenoids is that it really only produces a lot of the astaxanthin under white light and oxidative stress and so we really wanted to look at, understand how this pathway stimulation happens. So we built a light plate apparatus inspired by Jeff Tabor's work at Rice. And we grew the red yeast under different wavelengths of light and measured the fluorescence, which astaxanthin is for fluorescent. So it's a proxy for astaxanthin production under the different wavelengths of light. And we found primarily UV is the stimulant of carotenogenesis. And this is actually unsurprising. It's the red yeast is found on mountaintops. And it makes sense that it would produce a UV protectant like astaxanthin in response to it. So it's kind of a natural biological sunscreen for this organism. So we were able to determine that ultraviolet and pretty much ultraviolet alone stimulated carotenogenesis, which others hadn't found. But we wanted to ask the question, what part of the pathway is it regulating? And maybe this isn't entirely surprising, but we did find that the three terminal genes in the pathway are the ones that are most tightly regulated by light. So most of the upper carotenoid pathway shown here is used in a lot of different biological molecules like sterols. But those terminal three genes are necessary for carotenoid production, and those are the ones that are most markedly induced. And this was by qPCR. So we thought that there was a strong argument for looking at how light regulation was working in this yeast. And so we partnered with the Joint Genome Institute to do transcriptomics of oxidative stress and light. And we, based on the analysis of this transcriptomic data, we did find that it was reflecting what we saw in the qPCR data, that the 3 terminal genes, CRTYB, CRTI, and CRTS, are the ones that are induced when ultraviolet light is shown on the organism. And oxidative stress actually works on a more general level. It really activates almost all of the carotenoid and terpenoid pathway. So another question is, how might this be mediated? What's the mechanism? And we went and looked at a lot of different fungal photobiology that's published out there. And this is a figure that's adapted from a review. And we found that there are a lot of different protein classes, and we went and looked for homologs or potential homologs in the transcriptomic data, and we found that a lot of the cryptochrome homologs were being heavily turned on when we hit the system with UV light. And if you look over on the left, cryptochromes are UV blue responsive. And so this was a really strong case that we are identifying some of the first cryptochromes, some of the first photobiology in basidiomycetes that hasn't been seen before just with doing some transcriptomics. So that's a bit of the basic science. So we're kind of understanding how this light response works. And of course, we want to engineer it and take advantage of it. But there's another way we can use the transcriptomics data, and that is to get red yeast parts out. And we are able to, and what we want to do is have a modular cloning setup so that we can interchange any gene we want to express in the red yeast with any promoter or terminator. And we do that with Golden Gate cloning. Many toolkits are Golden Gate compatible that are out there, and we want to make one that works for red yeast. And so we went about this process of isolating lots of potential promoters from the transcriptomics data, looking for strong constitutive promoters, and looking for regulated promoters. So this is the strong constitutive promoters. So we took the top 10 strongest, highest FPKM counts from the transcriptomics that didn't really change under UV or dark. And we pulled the promoter sequence, cloned it into a synthetic construct, and integrated it into the red yeast genome. And we find that we can get most of those to be very strong constitutive promoters as well over a range. And so this really argues that we can, instead of trying to pull promoters from central carbon metabolism, we really can use transcriptomics as an upfront tool to derive parts. Many of these genes, besides ADH, are either hypothetical or not part of central carbon metabolism. The question is, can we derive light-regulated parts? And the answer is kind of a maybe here. We can derive, and we have found, a light-induced promoter that has quite a strong induction under UV light. But many of the other genes that were predicted to be regulated by UV, when we pulled them out of their native context, did not exhibit that type of regulatory behavior, whether induced or repressed. So we're able to get a couple of promoters we can use to turn on in the presence of UV in red yeast, which is really exciting. But we're kind of thinking about why this might not be working in every case, or why this doesn't work in most cases. And we're still that's still kind of an active area of research for my group. So finishing up the story on red yeast, what we've accomplished so far is we've determined that UV blue is the primary wavelength of light response. We've done whole genome sequencing and transcriptomics to really elucidate how the pathway is regulated and how light response might be working in this organism. And we've done a bit of domestication. I didn't talk about it too much, but we've done selection and transformation optimization and derived some modular parts that we can use to make pathways, maybe even deregulate the carotenogenesis. And we really want to understand why the transcriptomics poorly predicts regulatory behavior. And I'm noticing that I'm coming up a bit on noon. So I will try to get through this, but I think this is maybe some of the most exciting work that's coming out of my lab post-pandemic. We've been leveraging our knowledge of sequencing and modular parts assembly for these non-conventional organisms to harness emergent properties of the microbiome. And we really like to think about the soil microbiome, because there's this really diverse and complex interaction between plants, bacteria, and filamentous fungi in soil that really has been demonstrated to be, communication. And a lot of this is mediated by the fungal highways, and this term was coined back in 2005. So I use it to describe how those hyphae and those mycelium can transmit chemical signals and transmit information between plants and bacteria. And that's really what we're interested in. One of the things that people have noticed in the field and demonstrated is that bacteria associate with fungi for nutrition and migration. So they use the fungal mycelial networks to move in the particulate matter of the soil. And these are some microscope pictures we've taken of filaments and bacteria attaching along them. And that's really interesting. You can have something, an emergent phenomenon you can observe. And so we, being engineers and thinking about this, we really wanted to take advantage of this really cool emergent function that happens in the soil between fungi and bacteria. And we imagined that we could make this into an underground biosensor, where we could have fungi help the bacteria penetrate the soil and propagate the signal. And if we're able to have bacteria sense something at depth, it could tell us some information at the surface, so we don't need to dig for it. We use, again, a genetically tractable bacteria, Pseudomonas putida, to put a lot of our synthetic biology parts in here. And we use the fungal partner as a physical highway. And we're going to show you and hopefully convince you that it's necessary for the function of the device as well. Not only the genetics we're putting into the Pseudomonas, but the naturally emergent properties of the fungus. So there's actually several challenges here. There's a lot of genetic circuits, but not many are known to work in our bacterial host putida, Pseudomonas putida. Many are coming out now. There's been a lot of work on that in the field. So there's many more than when we started. Not a lot is known about how fungi might grow into soil, how fast they might grow, how fast they might transmit signals, which is one of the questions Alfonso was asking me earlier. And then how long they might persist, which is an open question. So my collaborator at WPI, Natalie Farney, has done a lot of the persistence work, and I refer you to her to talk about that. I'm really going to focus on the genetics we put into putida and a little bit of the dynamics of the fungi. So starting with the fungi, we isolated a species that grows really fast. And I'm going to move a little bit quickly through this just for the purpose of time. And it grows down into soil. So this is the bottom of a soil plate where the fungi grow right down in the center of the plate and then radially out. So we were really excited about this species to grow down into tubes. And so we made a bunch of columns and measured its growth rate. And essentially the fastest growth rate we found was in the largest column, up to 1 centimeter a day. The fungi can grow down into a 1 meter soil column. So we're excited about this species Elytratum for its ability to colonize the soil. Does it associate with Pseudomonas putida? And the answer is yes, it does accelerate migration. Our Pseudomonas can move at about 2 millimeters per hour down through a soil column, facilitated by the fungal highways. So we do have this emergent property that's occurring when we have a synthetic interkingdom soil microbiome. We screened a bunch of genetic circuits, and we found that the quorum sensing genetic systems worked very well in soil. And that's actually on the next slide, where we can induce them tenfold in soil when the fungi is present. And so this actually wasn't super surprising, because quorum sensing does work in, is derived from pseudomonads, but we were able to, again, demonstrate that it works, particularly in soil with the fungal partner. And I emphasize that because this is not an LB, this is not in rich media, this is not at exponential growth phase, and the genetics are working. So how do we use these? We've been able to engineer the bacteria. We show that they can work with the fungal partner. How do we use the system to propagate the signal? So the first thing we did was design a positive feedback loop that would help us propagate the signal to the surface. And then we can also boost that chemical transmission of signal by adding a chemo attractant to facilitate bacterial migration. So I'll finish by demonstrating both of those. So the quorum sensing feedback circuit works by making the LuxR protein constitutively, and in response to one of the homoserine lactone inducers of the system will activate the promoter, either driving RFP for a signal or production of LAS-I, which will generate more homoserine lactone. And so you get that feedback, positive feedback circuit. And we also have constitutive expression of GFP to detect cells that are engineered but not turned on, so not making RFP as well. So I'll show you the data of how this works. So here is our soil column. We have the genetics in the Pseudomonas putida, and we're not inducing at this point. We're only adding the DMSO solvent. And what we see over on the right, performance in soil without fungi and performance in soil with fungi. The y-axis is the height in millimeters along the column, and the x-axis is the time in days post-induction. And so you can see that really we're not seeing many positive cells in any part of the column over time. When we hit the system with the quorum sensing homoserine lactone inducer, we do get activation of the system day one without fungi, but it quickly goes away. But that emergent interaction between the fungi and bacteria allows the signal to travel further and also last longer over 5 days. And so we're still getting positive cells at day 5. We can change up the inducer we use, and we can send the signal a little bit farther. In this case is 6 centimeters. The other thing we can do is switch out some of the promoters and redesign the circuit. And so this is with a different design. It's a little bit leakier, but it sends the signal much farther. We can send the signal 8 centimeters and have the cells display a positive signal over 5 days. And so this is really exciting. We're using those fungal highways, and they're clearly important for this type of communication, quorum sensing communication between the bacteria. And we can send it up through a soil column from depth to the surface. Another thing that can help us do that, I mentioned previously, is the chemo attractant. And this is just chemo attractant alone can move bacteria up through the soil at about 1 millimeter per hour. When we combine those, what we're seeing here is, again, similar data of what we showed before, but water and salicylate. And what we're showing here is that we can, with the most important part here is the bottom right, everything else is more or less controls. When we hit it with the inducer and the salicylate chemo attractant, we can get really rapid induction of the genetic circuit, propagation through the soil column, and the signal lasts for days with many positive cells present even a week later in the soil. And this is key to have the fungal partner. If you don't have the fungal partner, the signal doesn't last as long and it's not as persistent. It doesn't go as far and it's not as persistent. So finally, we've established an interkingdom genetic device in soil by combining the emergent properties of both bacteria and fungi. And I think this really speaks really strongly about why we should be working in non-conventional organisms in unconventional conditions, because we can benefit from these emergent useful phenomena. And who knows what we can build from here. There's a lot we're thinking about, and there's a lot that I think others could think about. When you look at the microbiome, you look at nature, you look at how ecosystems are organized, and really think about how we can use synthetic biology to affect that for devices that are useful and maybe even beneficial to the environment. So with that, I'll end. And I just want to have a brief note on the research accelerator we built at WPI. I advertise this every time I give a talk. So if you are in the Massachusetts area or not, and you would like to access some of the synthetic biology tissue engineering and gene therapy tools that are really accessible to a lot of highly funded centers, you can access it at WPI as a fee-for-use facility. Please reach out to me or series@wpi.edu for more information. And with that, I'd like to acknowledge all of the funding sources that made this possible and all of the great people whose effort made this possible, particularly over the pandemic, where we've made progress on a lot of these different elements. So with that, I'll take questions. So thank you very much again for giving me the opportunity to speak today.
Amazing talk. Thank you. Thank you. You know, that you giving always the great talk. So so it's fascinating. I see Alfonso's hand, and you can speak. I mean, to ask question or comment.
Okay. So and thanks, Eric, for this very nice talk. I mean, very stimulating. So I have a few questions. So I don't know. So let's start with the most pressing one, is that about these fungal highways. So if you have intermediate concentrations of your, I mean, a signal, I mean, OC6 AHL, and will it get transmitted also to the same intermediate concentration, or because of the positive feedback, it will just get propagated to the saturating level?
Yeah, that's a great question, and that's something that we're following up on. It would be great to have it be kind of a sensor for the concentration you're encountering in the soil, but I do think that by incorporating the feedback circuit for us to help propagate the signal, we're kind of going to be more of a digital response. So if you do detect some, you would get the full signal. We haven't demonstrated that yet. We really, again, we're trying to prove that this works. So we were really putting a fair amount to induce it at the bottom of the soil column. But yeah, that would be something for us to look at.
Because for me, it would be very nice not to have the positive feedback at itself, so to have this graded sensing response, because that would allow you to enter to the analog world, and then I mean, you could do so many things. I mean, but I understand that as a first step to have this in a Boolean type of behavior is the first step. I mean, this is new. So then, I mean, I could never expect to have this through the soil. I mean, having this communication, I mean, to happen, which is already, as it is very, it could be very useful for my bacteria when I was talking to you regarding the having a communication channel. And currently, what we are doing is we just print in agar plates the channels. Okay, so but it would be good to have in the soil the channels with fungi, and then bacteria communicating with each other and doing some neural computation as we do and using your highways.
Yeah, I think that that's what we've demonstrated here, is that the fungal highways, we can co-opt their natural ability to transmit chemicals and move bacteria to send synthetic genetics signals. Yeah, there's the applications of this, I think, are very broad and diverse, and I'm glad you're thinking about other applications.
Yeah. So I think that to my mind, yeah, because I mean, this is the core thing of signaling, and then signaling in the moment you you make in in micron distances, but you have the really, I mean, where you don't have the decay of the signal, because the big problem in when you do the normal quorum sensing is that it diffuses everywhere. So then you need, you are wasting all the chemicals.
Yeah, and that's what we show. I mean, without the fungi here, the diffusion just kills the ability of the circuit to send the signal any distance.
Yeah, that's very nice. I have just a technical question. I mean, just particular question is, what are, because when you show the sequencing data, I mean, and then you show the in this plasmid with I saw 25 copy number. What is the highest copy number you can have in yeast?
That's about, so for the that's a 2 micron plasmid, so that's about the highest you get. there are reports of that 2-micron plasmid being up to 100 copies. We measured it at about 25 in our work, in our hands. And that's within the range of what you expect. And it's like a lot of high copy plasmids where it's highly variable. So the number can vary widely. So it's 25 to 100 is generally what you find people reporting for that copy number.
Do have people really re-engineer the copy number control to make a higher copy or lower copy in yeast with these 2 micron?
So I think that there's more effort in simply integrating multiple copies into the genome, since it's so easy to do that in SynBYSS. There hasn't really been much on manipulating copy number of those plasmids.
I see.
Yeah.
I mean, I'm a work all the time with plasmids. So then I always think, I mean, in fact, we are using an analog memory based on plasmids. So then if I can use a plasmid from yeast, that I can, then the capacity of the memory is given by the copy number. So there is a incentive to do that. And if I use genomic copies, I cannot tune the memory.
Yeah, so you do have some flexibility between the low copy number, which is 1 to 2, and the high copy, but not really much in between. And not really much control over whether it's 25 or 100, as far as I'm aware.
I see. Very good.
Yeah.
Okay. Thanks very much, Eric.
Thank you, Alfonso. I mean, this is a great questions. So actually, we already passed the 11, my time in the 13, but still I want to ask one question. So could you share your wisdom regarding how to enter fungi synthetic biology for brave younger people than you, or including myself, because I never imagined to enter the fungi. So what would you do differently if you start the project again, right now? What would be the remaining key challenges or question or problem, etc.? Although you already gave a lot of, many challenges, I mean, in this fungal, the synthetic biology field.
Yeah, so the biggest hurdle that you have to get over when you start working in fungi is keeping, and you're from the bacterial world, is keeping in your mind that again, like those different solar systems I showed, they all operate on the same basic principles, right? They all operate on physics and gravity. But so they're tied together by the central dogma. Transcription and translation exist, but the molecular biology of those things are very different. And so if you look at how we can, a lot of repressors can transfer fairly easily between prokaryotes and eukaryotes, but that's the major division is just taking some time to learn eukaryotic molecular biology, and that can give you a foundation to move very comfortably into fungi. That's like the technical answer, and just the motivation for moving, I think, is kind of shown in hopefully these vignettes that I've shown here, where if you get excited about I always tell my students, right, I don't really care about your background or where you've come from or what you've done. If you're excited about what you're working on, you're gonna learn what you need to do. You need to get to do what you want to do. And so if any of these things captures your imagination in the way it has mine, then I think you're well on your way to entering the deep end of fungal synthetic biology.
Right, exactly. So that's kind of resonate with me, because I just gave the talk, for the ONR program review, and there's I would say the craziest idea I ever imagined. And then when my student actually approached me saying, I want to do this project, and then I said, are you sure? This is this is a very, very challenging one. I even do not I'm not sure whether this is working or not. And then, do you want to invest your time? And then she said, yes, why not? And then she actually made tremendous progress. And then I really glad I report today and confidently, I could, submit this paper to Nature or whatever Nature journal, and confidently I said that, and very proudly. And then same thing, but I still, cannot touch the fungi, because fungi is fungi. I mean, this something crazy guy or fungi, whatever. So yeah.
So the other answer to how to enter the fungal world if you're young and starting out is to come join my lab.
Yes,
that's
yes.
the other answer.
That's the simple answer. Yeah. So if you wanted to do fungi, I mean, just join Eric. That's wonderful. So I'll double check whether we have the question from general audience, probably, but probably everyone let me check. So one more time, Q&A. Okay. But we already have the interesting discussion, so they already kind of enjoyed that one. Okay, let me close, because we should kind of be mindful of the time. So thank all for joining and staying today, although many things are going on, including the most interesting, exciting synthetic biology conference, SID conference currently happening, and also final exams. So we will meet again next week on May 12th, Thursday, the same time, the same Zoom link. But I will be joining from my home temporarily before I start my 70 days of seminar trip worldwide. We'll have Professor Matthew Chang from National University of Singapore. I believe at his time, probably 11 p.m. And Professor Lakra Bintu from Stanford University, where I will actually visiting to give my first seminar for the 70 days of non-stop trips. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty face. Thank you, and I'm stop recording. Let me stop recording.